What have we got? Oh, 29.
Sorry.
I was looking to see how many had joined so far.
Yeah, 29. We've got about, I think, 40 people. Ella said about 40 people that have registered. So while we're waiting for the rest to join in, why don't we just pick on 2 people in the room and get them to introduce themselves and just tell us why you're here today. So why don't we, Lance, you pick on one.
So I'm just changing to gallery. I'm just going to pick a name that I don't know, which is very easy with most of them who are on there. I'm just going to go Jamie Attenborough.
Okay. Jamie, are you there?
I am here. Sorry, I am here. I had a piece of apple stuck in my throat. I was eating some apple, so it's great timing, Lance.
That's the way I roll.
No, that's all good. Yeah, so my name is Jamie Attenborough. My camera should be loading, and I work with Di and the team on the commercial translation module with the TTM, but I also work with the Health Day Activator at Callaghan Innovation, which is New Zealand's government innovation agency for the Australians joining. So thank you.
Thanks, Jamie. Okay, so my turn. How about Masako Dan?
Sorry, trying to. Hi, my name is Masako Dan. I'm from actually Sydney, Australia. I work with the head and neck surgeon, Jonathan Clark. I'm the research and innovation manager at the head and neck at the Chris O'Brien Limehouse. So it's a cancer hospital that's associated with RPA hospital. So we work on 3D printing, tissue engineering in collaboration with Gordon Wallis from Wollongong University. So yeah, just quite keen to listen in and what you guys do, and yeah, that's why I'm here.
Aidan, do you know Masako?
Sorry.
So do you and Aidan know each other?
Aidan, no.
No, I don't think so.
Yep.
We probably
We should
know each other by proxy with Gordon maybe, but
Yeah.
no, I've never met Masako before, I don't think.
Yeah.
So thanks, Masako.
No problem.
Great to meet you.
Nice to meet you too.
Okay. I guess shall we start? We've got a quorum.
We'll make it that.
Yeah. Okay. So welcome everyone to the Australia New Zealand BioBridge workshop on 3D printing in health. My name is Diana Siu, and I'm the co-lead of the BioBridge from the New Zealand side. And I'll let Lance introduce himself in a little bit. So the BioBridge was actually initiated to try and link together New Zealand and Australia's health tech innovation ecosystems. And we try and support and make connections between researchers and clinicians as well as industry. And what we've also been doing in the last couple of years due to COVID, because no one could travel, is actually have these virtual workshops where we pick a topic of interest to the community and have a little curated workshop as with respect to the topic, and invite the people that we think will be interested to come along. So I'm based out at Auckland, at Auckland University of Auckland, at the Auckland Bioengineering Institute. And I also lead some of the national programs that underpin research translation. So Lance, I'll hand this to you to intro yourself.
Yeah, thanks, Di, and good morning, everybody. So I said, my name's Lance Chia. I'm the Director of Investment and Innovation at the Liverpool Innovation Precinct. There's 10 institutions in that initiative. There's 3 universities, Southwest Sydney Local Health District and the Ingham Institute for Applied Medical Research. Just mentioned 5 of them. And as Di mentioned, the purpose of the BioBridge and these workshops is to really try and connect researchers and people and ideally develop some collaborative work. And this is the fourth of these workshops that we run, and we've had some really, really strong and valuable collaboration and work come out of previous workshops. And so, the reason I mentioned when Masako introduced herself to Aidan, because I know a little bit of Aidan's work, and if I can recognize some places where people might have some common interest, then we'll curate those where we can. So through the workshop, if you know someone or you don't know someone and there might be something of interest to you, certainly stick your hand up, and Di and I will help connect you, help you kind of get on the same page and see where that might go to. So yeah, thank you for joining us all today. I've personally been really looking forward to this one. Koen knows how interested I am in this space. I'm aware of the great work that Aidan and Inventia Life Sciences has been doing, and I was looking forward to meeting Seamus as an all-new person. So look forward to getting going, and yeah, thank you again for joining us.
Okay, so so today we've got 2 Kiwis on the panel and an Australian. So welcome Koen and Aidan and Seamus. Koen and Seamus are from New Zealand. Aidan's from Australia. Now, the panel that we've put together today will cover all, research translation all the way to commercialization. And Koen represents that early stage research translation side. Koen is a researcher at the University of Otago, and he's also co-leading a couple of the programs with myself around research translation. So I'm sure you would have read their bios, so I won't go any more into that. And not to waste time, I'll hand it over to Koen. Now, what we will do is we'll take 1 or 2 questions just after each panel speaker, and then we'll hold the rest of the discussion for later, okay? And if I can ask everyone to keep to time, Koen and Aidan and Seamus, that would be fantastic. Okay, over to you, Koen.
Thanks. Can everyone see my screen?
Yep.
Awesome. Thanks, Di. Thanks, Lance. And also Kate for the organization. Thanks for having me. And I think what I want to show today is a bit of the work that I've done here, along with Tim Woodville at the University of Otago Christchurch, and on the quest of what we have done in the last 8.5 years or so in developing some new bioinks. Specifically, we work with bioprinting, so it's really looking at printing with cells in these materials. So not just 3D printing, we look at bioprinting, and then a little bit of the focus on what we are doing in the group as part of the Department of Orthopaedic Surgery and Musculoskeletal Medicine, and a bit of the vision of where we see how bioprinting or biofabrication can contribute to research, but also to get into inpatients. So I guess we all know that there's a lot of 3D printing technologies out there. There's a lot of bioprinting technologies, biofabrication technologies, and these are really just a few that we have set up here in the group here in Christchurch. We do a bit of bioassembly, lithography, extrusion-based bioprinting. And I think the real take-home message that I hope to get across to you guys today is really all of these technologies have their pros and cons. They have different resolutions. They can make different shapes. Either some can do multi-material, some can't. So it's really looking at what application you are interested in, and then you sort of pick the right bioprinting technology that allows you to reach that application or that criteria that you actually want. So your output. What we do as part of the Department of Orthopaedic Surgery is really looking at printing the joint space. So we're looking at the bone and the cartilage and that interface, how to get these constructs integrate really well with the host patient. And I'll just show a quick video here of sort of the vision that we collaborated with Professor Jos Mauda here at WMC Utrecht in Netherlands, where really the idea is looking at if you have a patient with an osteochondral defect, you can have the defect scanned. You can translate the information to a 3D bioprinter. You can instruct the printer to print, different materials. And in this case, we're looking at a mechanical support structure, and you have different bio inks in them. So you can have a bone bio ink, so a material that supports the cells. You can put growth factors in them, different proteins, ECM matrix components in them, and you can start printing this in a layer by layer fashion. So in this case, we started with the bone layer, then the deep zone layer, middle zone layer, and then the superficial zone layer for cartilage. So you have sort of an osteochondral construct that you can transplant back into the patient. So a lot of the work that I've led within the group is looking at making these different kinds of bio ink. So we use hydrogel, so just this jelly-like, highly hydrated polymeric substances that we know that we can put cells in them. And our choice of material is actually just gelatin. So, it's easily accessible. Gelatin is well known. It's actually very well known to be cell-friendly. It's a form of denatured collagen, and its temperature sensitivity gives us, the additional benefits of this to extrude this material. But also, as a polymer chemist myself, I do a lot of chemical modification, and gelatin has a lot of this end groups or pendant groups that allows me to modify with a whole bunch of extra chemistry on it to make our bio inks more special. So our base material is gelatin, and we often modify it and then combine it with a visible mediated cross-linking system. Now, I don't really want to bore you guys with all of the details of the chemistry, but we do have intellectual property around the use of this visible photo-initiating chemistry in terms of making these bio inks printable and then photo cross-linkable. But what I want to draw your attention to is why we are interested in using light. So photopolymerization gives us spatial temporal control over the cross-linking process. So this is just an example to show we have a photo cross-linkable polymer, in this case of a bio ink. We project a Mickey Mouse pattern into it. Wherever the light hits is where you get a gel. So you can very clearly see that you get a Mickey Mouse hydrogel printed now on the slide, and you can have uncross-linked polymer that just wash away. And really, if you can control this properly with the right resolution, you can sort of try to make Mickey Mouse dancing on the glass slide. So the photopolymerization technique gives us the spatial temporal control. And what's really unique with our visible photopolymerization system is that we know that it's really accessible. We've tried cross-linking just using the torchlights on phones. It's visible light, it's very cell-friendly. We've tried a range of phones, so we've definitely tried the Samsung, the Huawei, the iPhone, and it all works. So that is sort of the basis of the technology that we've developed. So going more into the specific kinds of bio inks that we make, if you can remember, I spoke about the osteochondral construct. So we look about, the cartilage and the bone layer. So at much earlier stage of the research, we focuses on using the gelatin-based hydrogels, try to grow articular chondrocytes within them, and we find that you have to biofunctionalize this gelatin-based bio ink. So in this case, we added heparin into the material, and you can here very clearly see that with and without heparin, you have a lot more tissue that's being formed. So the cells are much happier within this hydrogels when you have heparin mixed into the gelatin. And we can also look at culturing them in different kinds of environment. So in this case, we look at a normoxic versus a hypoxic environment, knowing that cartilage is avascular, aneural in physiological environment. So by not giving the oxygen to the articular chondrocytes, you can see that we get a huge, a lot more amount of tissue that's being formed. So that gives us a little bit of, different kinds of ways to control how these cells are going to behave within these tissues or tissue constructs that we make in terms of engineering more functional construct. We also spend a lot of time making bone bio inks. Let's talk about cartilage bio inks. So we take gelatin, we mix with heparin. For bone, we take gelatin, we mix with strontium nanoparticles. So we make these nanoparticles. We use strontium because strontium ranelate is really a well-used drug used to treat osteoporosis. So you can mix the strontium nanoparticles into the gelatin. You can print them. It stays pretty stable over time. So from day 0 to day 28 days through cell culture, you can see that the fibers stay stable. But over time, you can see more of these different black specks within these hydrogel fibers. And when you stain them, it's actually the mesenchymal stromal cells within the fibers actually starting to mineralize. So they're depositing down the calcium around it when you stain it. So you can see that the presence of the strontium actually then supports the cells to then start making bone within this printed construct. We also look at vascular bioinks, so cartilage is avascular. For bone, it often is vascularized because you need to get adequate nutrient and oxygen supply to bone to keep it happy. And in this case, we also made a range of gelatin-based hydrogels, but with different compressive modulus. So the mechanics or the stiffness of this hydrogel is actually really important, where we found that a soft hydrogel cells are able to form microcapillaries, but if the gel is slightly stiffer, the cells don't do that, so they just sort of migrate to the surface and form an endothelium. So it is not that simple to try to recreate the native vasculature system where you have sort of a macrovasculature to a microvasculature. I mean, we're on our way there. We're really trying to do that. But what we find is that it's really important to co-culture cells within this bioink. So in this case, we had to put endothelial progenitor cells and then stabilize with human mesenchymal stromal cells, because the stromal cells, which is the red here, comes and stabilize the microcapillaries that are being formed. So besides just the material, the interaction between the cells is also really important in our quest to make these functional constructs of printing these constructs. So that is a little bit of our work around the osteochondral space and using gelatin-based bioinks in the visible black initiating system. We also work with other bioinks. This is specifically what actually Lance wanted me to show a bit of a DECM work. So what we have done here is that we took cornea and we took heart from a pig. We decellularized it, so we remove all of the cellular components of it, and then we mix it with our visible light photo initiating system and use it for 3D bioprinting. And the reason why we needed to photo cross-link it or photopolymerize it, because it gives it additional mechanical stability. So native decellularized extracellular matrices will just collapse. Once you photo cross-link it, you can see that you can start stacking them into multiple layers. So it's a lot more stable. And because it has this additional mechanical stability, we can make bigger macro tissue. So in this case, we can print a human-sized ear, much bigger constructs. But what we also found that was interesting is that the mechanical robustness of these materials are actually quite good. So you can make them into a disc, fold them into half, refold them into half, and they actually don't break. Or you can also print these really large fibers, and you can also start twisting them, and they also don't break. So the mechanical resilience of these decellularized extracellular matrices now are pretty good. In terms of function, so for example, we look at cornea. So because now we have the additional mechanical stability, we are able to print it self-supporting, so you can print a concave structure. Once you immerse this into PBS, I think I'll draw your attention to the T that is in the middle of the cornea. You can see that over time, the T gets clearer. So the optical transmittance of light through this bioprinted cornea is close to 100% to show that it's actually functional. You can use this as a substitute for cornea in this case. And we also spend quite a lot of time trying to make these bioprinted heart models. In this case, we could induce pluripotent stem cells into them and look at the retention of some of the cell phenotypes. So cardiac troponin, connexin 43 in this case. Recently, we have expanded this to decellularized liver, decellularized colon as well. So there's quite a lot of extra work that's coming out from this. But even thinking about trying to make a functional tissue, I guess what I want to allude to this is it's actually quite difficult. It's not as simple that having a material and having a cells and you just mix them. So in this case, the priming of the induced pluripotent stem cells to make them into cardiac myocytes is not that straightforward. And there's a lot of biology that goes into it to make sure that it retains its phenotype. But it is really rewarding once you see that the cardiac myocytes are able to be in the printed construct. In this case, where we had to mix the cardiac myocytes along with cardiac fibroblasts, print them in the decellularized heart bioink, and then leave them for the spontaneous beating motion. So now we have these different mini heart tissue models that we can use for a range of downstream applications. So we've captured some of our vision in here where, how nice would it be if at some point we're able to have a printer that would allow us by the tip of our finger can print a range of different, organs. That's sort of where I see the field is moving towards. Have a couple of minutes left. So What we also have been doing with a lot of these bioinks is looking at making microfluidics model. So in this case, we printed a range of hydrogel chips. Particularly, this is interesting because this is an angiogram that we took off a patient, which has the truncated blood vessel here, and we're trying to replicate that in the printed construct here. You can see that you can have two open channels that are perfusable, can have two kinds of liquid perfused through it. We also look at some cancer models. I again won't go into too much of the details, but in this project, we're looking at capturing the cell signaling between breast cancer cells and mature adipocytes. And by just drawing your attention to this panel here, where the presence of the adipocytes do cause some sort of EMT transition of the breast cancer cells. So in terms of trying to use this as cancer models, it is important in terms of a breast cancer model that you also recapture the interaction between the cancer cells and mature adipocytes, because in the breast environment, the mature adipocytes is quite abundant, and there is a lot of cell interaction between them. We also look at some vascularized cancer organoids. In this case, we look at, building in microcapillaries within these printed constructs of cancer cells. You can see that the endothelial cells standing green here, once they're in contact with the cancer, they start disrupting, they lose their phenotype. So cancer is affecting some of this vasculature, and it's quite nice to be able to capture this in our printed model as well. Just a little bit of cancer models that we've done there, a bit of gelatin-based bioinks, decellularized extracellular matrices-based bioinks, some of the models we have done. But recently, we're also moved away from just making the materials. What about inventing or building our own 3D bioprinters, that are much more accessible? And in this case, we were able to build a mobile phone-based DLP printer completely just using a smartphone. Wrote an app for it, so I'll just show a quick video showing how everything was just done on the smartphone where you have an app. For example, this time we're trying to print a gyroid. You can slice it into different layers, and this is the printer. This is our print platform, and by just controlling, intensity, speed, everything on the app, we're able to then have light projecting, printing the material all on the base bit, all done just by using a smartphone. So looking at making this a lot more accessible, and also our vision moving forward is that potentially at some time we're all able to just use smartphone, scan a defect, translate everything into a printer, completely operated by a smartphone. Everything is just digitalized in the hospital, and how great would that be? So I'll just end my talk with just also declaring conflict of interest that we do have an intellectual property. We have a full utility patent now granted in US, Europe, and also China. And we have licensed some of our bioinks to a couple of companies. So there's a couple of kits out there using our technology. So we have done a little bit of research commercialization as well. And I'd just like to acknowledge the team, all of our funding agents, our collaborators that help participate in a lot of our work and making this possible. And lastly, invite you guys to come to our next Australasian Society for Biomaterials and Tissue Engineering Conference that will be hosted in Christchurch, 12 to 14th April next year, that I'm chairing. Looking forward to see all of you here in Christchurch, beautiful, beautiful city. And happy to take any questions. I've also have our emails here, so can either contact me or contact him, and we'll be more than happy to answer any questions you have related to our work. Thank you.
Great. Thanks, Kun. That was really interesting. How far have you got with your mobile phone app and 3D printer?
We looking a lot more into optimizing the materials that can be printed with them at the moment. So what we published was our first prototype, and we just use a couple of materials. But at the moment, we're looking at also trying to improve the resolution, the printing. So we know that it's doable. We know that we can print fast, but by printing fast, we're also losing resolution. So we're trying to look at how can we then, improve the resolution without diminishing the printing speed as well.
Probably a conversation with Aiden might be good. Yeah. Okay, open to questions. Does anyone have any comments or questions for Kun?
None.
No?
Very
Looks-
quiet. Yeah.
Okay. Well, we'll leave that till later, but looks as though you've bamboozled everyone, Kun, but thank you. Okay, Lance, over to you.
Yeah, thanks very much for that, Koen. Yeah, it's my pleasure to introduce Dr. Aidan Mahoney, the co-founder and chief technology officer at Inventia Life Sciences, which is I think now a pretty well-known and successful scale-up company here in Australia. Aidan was part of the development of their first product, which I'm going to guess a few people on this workshop are familiar with, the 3D bioprinting platform product called the Rastrum. I don't know if he's going to talk about their new project, but at least now we know he's focused on translating that core technology to the clinic, and in his words, transforming human health and improving patient outcomes. So stepping outside of the research lab. And so, yeah, thanks for coming along and joining us, Aidan, and I'll hand over to you. Sorry, my dog.
Thanks, Lance. Thanks for the invite to present, Lance. Just figuring out here if I can get my tab to share with everyone. Everyone can see that? Yep.
Yep.
All right.
Yep.
Well, yeah, thanks, Lance, for the invite. It's really nice to be here to talk to you a little bit today about Inventia, and in particular LEGO, which is an in situ bioprinting, I guess, product that we're developing for treatment of full thickness skin wounds. So, as Lance said, my name is Aidan, one of the founders and the CTO at Inventia, based here in Sydney. I'm not actually, well, I am Australian now, but not originally. I'm originally from Ireland, so just in case that's confusing anyone with the accent, I'm not originally from Sydney, but Inventia is based here, and that's where we do all our development work. So maybe before we get into LEGO and all of that exciting stuff, I'll maybe talk a little bit about Inventia in general and about our mission. So it's pretty simple. Inventia's mission is to be able to create human tissue at scale. So we want to be able to create little pieces of brain tissue, liver tissue, skin tissue, whatever tissue you want. And we want to be able to do that in a truly scalable way. So to make a big impact on health and making it possible for any scientist or clinician to create human tissue on demand. So that's what we've been doing since 2013, since we founded the company. And we'll hope this will drive a major transformation in medical research and health in 3 main areas, so that we've been working towards and building on each of these different areas. So the first one is disease research, where we've got incredibly, smart scientists, and there's probably some on the call here already, fighting, cancer and other diseases, but stuck with old tools like cells growing on plastic that doesn't really give them the insights they need about whatever disease that they're interested in or looking to develop new treatments or cures for. So we're trying to enable better research in 3D tissue models that mimic human biology more closely. The second area that we're working on is drug discovery, where the way that new life-saving drugs are developed is still really inefficient, all because the vast majority of drugs that look great in lab testing end up failing in people and in clinical trials further down the drug development pipeline. So we're trying to change that paradigm, hopefully by enabling more predictive testing that's done in human tissue. And most importantly, that's done outside of the human body. So trying to enable that for the folks working in drug discovery and development of new treatments. And then finally, the third area that we'll talk a little bit about today is clinical regeneration. So this is something that we've wanted to do from the outset of the business. This was always the long-term vision, was to build on those first 2 horizons and develop our technology and use that to translate it to clinical applications. So we see a future where, you know, patients' own cells can be used to regenerate fully functional tissue and organs. And we think this is now within reach for some of the simpler tissues like skin. And we're already kind of printing new skin tissue because we're kind of building on those first two horizons and the technology that we've already developed. So what is our core technology, I guess, and how are we translating that to the clinic? So Inventia has developed core technology in three main areas. So we use a droplet-based bioprinting approach. So a little different to what Koen mentioned earlier in his talk. I think majority of folks are using like an extrusion based, but we use the little micro droplets, which we put down, allows us to put down our biomaterials in a really scalable and kind of more high throughput manner. We've got our own printable matrix materials that we developed, predominantly a PEG-based biomaterials that we can use. They're in liquid form at room temperature, and we can use them with our droplet-based printing system to create hydrogels and extracellular matrix mimics. And we can also modulate things like stiffness, for example, with those biomaterials. And the final piece to that is tissue relevant structures. So putting the first two together, we need to be able to create tissue relevant structures that will be able to form, you know, tissue essentially, either in the lab or you know, in the clinic. And so we've been busy, I guess, trying to commercialize this core technology into our first product, Rastrum, which is this very shiny pink and chrome lab instrument. So this is really targeted at for disease research and drug discovery. And we've already support many different tissue types with Rastrum. And this is being used around the world now, which is very exciting. We've got many customers here in Australia. We've also got customers now in the US and Europe that are using Rastrum to kind of power their early stage biomedical research. And then the other, I guess, development that we've been pursuing is in regenerative medicine, where we've been using the same core technology to power LIGO. And we want LIGO to help us to change how, you know, tissues are regenerated in the clinic. So the first tissue type that we're targeting is skin, because we feel like that's, you know, a relatively easy place to start.
Hi, Mum, you all right?
Sorry?
Darling? You all right?
Oh, I can hear you. Yeah.
Can we just ask people to put all their devices on mute, please?
Thanks, Lance. But we do see a future where we can use LIGO to target other tissues other than skin, cartilage, for example, and many others. So we're developing it with that in mind. So what is LIGO? Well, LIGO is a platform that's going to improve the standard of care for full thickness skin damage. So currently on the market, there's a number of I guess approaches you can use to treat full thickness skin damage. So you can have skin grafts, which are probably the most widely used. So a split thickness skin graft, for example, that needs to come from a donor site on the patient, which I've been led to believe can often be more painful than the wound itself. You can use cell suspensions without a scaffold. So they seem to work pretty well for wounds or skin damage that is not very deep. So when you get into the deeper hypodermis or dermis layers, that approach doesn't work very well because without a scaffold to keep everything in place. You've also got cell-free scaffolds. So there's many of those available in the market, and seems to be more coming onto the market these days. So they can be animal derived or synthetic. But I guess what the drawback we see with these is they don't really mimic the native tissue because they don't have any cells to kind of trigger that regeneration. And also existing standard of care requires multiple procedures, and that's a really increases hospitalization costs, and it's not a very nice experience for a patient. So what are we trying to do with LIGO? Well, we want to basically unlock the regeneration for full thickness skin damage. So we're not really trying to print skin here, if you want to think about it that way. What we're trying to do is to put enough biomaterials and cells into a wound site that will trigger regeneration and trigger that, stimulates that regenerative phenotype, and it will promote tissue-guided regeneration. So essentially to reduce scarring and speed up how fast the wound will heal itself. And we want to do all of that in a single procedure, and we want it to be customizable to the patient. So for example, we can customize to the depth of the wound, the size of the wound, and we want to be able to do that in kind of a highly scalable and adaptable way. So we want to be able to use LIGO not only for skin, but also for many other tissue types in the future, we hope. But right now we're focused on skin. It seems like a good place to start. And from there, we can hopefully leverage our experience and focus on other tissue types at a later time. So how do we see LIGO working? So the first step is really harvesting the cells. So you need a cell source. So you can do this in kind of two main ways. You can get cells from the patient, so autologous cells taken from a donor site. So this is done with RECELL, that kind of spray-on skin product, for example. Or we could have cells from an external source, so allogenic or established common cell lines. So this is becoming more and more common to have these established cell lines available commercially off the shelf. And then the second step is we combine that cell source with our biomaterials, which have been optimized for whichever tissue, in this case skin. These are PEG backbone optimized with peptides and proteins that are native to the body to again promote that regeneration. And then the final step is basically we load all of those biomaterials into our drop-on-demand printing platform, and we deliver those directly to the injury site in the operating theater. So this is the kind of workflow that we are trying to target. So and we're trying to keep this down to about an hour. So this is we've already been doing preclinical studies using a LIGO prototype. And so again, the steps, we've got a setup time, we've got preparation of our cell source, which in this case is RECELL, so it's an autologous cell source. We've got to load the bioinks into the device. Got 30 minutes of printing time where we print directly into the wound site. And then finally, we've got the dressing step so that the wounds are dressed with a standard wound dressing. I'm running a little bit over time, I think, so I'll got 2 more slides. So hopefully it won't take too much longer. So as I said, we're in preclinical trials, and we've got some pretty, promising looking data so far, and we've been looking at this both in vitro using rastrum, so we can look at this in the lab, we can optimize our biomaterials, and we can basically try and reduce the list of candidate biomaterials for use in the in vivo studies. So there's still a long way to go. This is like pretty early days, but we are targeting kind of a first in human feasibility study probably at the end of next year. But we've got quite a bit of work ahead of us to kind of build up all that data and the scientific foundation to get us there. Finally, this isn't just all of Inventia's work. We've assembled a pretty amazing team across Australia. So we've Professor Fiona Wood is our clinical champion who's really been helping us from the outset. Fiona is very passionate about treatment of skin injuries, in particular burns. You know, she's dedicated her life and her career to that. Also got Professor Gordon Wallace. I'm sure many of you will know Gordon. So he's you know, doing incredible work with his team at the University of Wollongong into advancing biomaterials. And then also we've got our consultants who've also been helping us with different aspects of the project. We've also had incredible support from the government in terms of funding, so the Biomedical Horizons grant and also the Medical Device Fund. So we're incredibly grateful that we've had all this support and help to try and make this device a reality. So that's it. Happy to take any questions. I hope that gave you a little bit of an insight of what we're trying to do here at Inventia and our mission.
Well, thanks for that, Aidan. Actually, you've progressed quite a bit since the first time I heard about what you're doing with LEGO, so congratulations for that. I think in the interest of time, anyone who's got questions, we'll save them for the end. So we'll move to Seamus, and then we'll all come back together for really just an open Q&A under a panel type format, if that's okay. But yeah, thanks very much, Aidan.
No worries. Thanks, Lance.
And yeah, over to you, Di, or Seamus.
Seamus, we'll go straight to Seamus.
Great, easy. So I'm Seamus Trudinick. It's kind of nice to bookend my talk with Koen's talk. I guess it's the what I call the blue sky to blue ocean sort of journey from getting something out of the lab and I guess into that mundane, you know, established technology market that we call commercialization. So I'm just going to share my screen with you here, and hopefully we can make this work. Give me just a second. So I'm going to give you a little bit of who we are and sort of how we got here. My role at Ostability is to help vets do better surgery. And the way that we do that is by providing primarily additive manufactured implants and surgical guides. I'm an engineer by training, and so I'm a lot more focused on the applications of this technology rather than the nuts and bolts behind it. But this is really a story from my discovery of the potential of AM through that commercialization journey to where we are now, which is growing, but we've got a long history behind us. We've now treated over 5,500 patients. So it's maybe a little bit different to some of the other talks that we've had. All right. Have we got my screen there? Maybe not. Which screen are we? Can anyone tell me if we can see?
Yep.
Yeah.
Yeah,
Okay.
can see it now.
Right. Cool. So in 2007, I did a summer internship with a local manufacturing company. That company was called Instech. And I guess I was in the right place at the right time. We had some friends over in the US that had just put in their first, actually the first RCAM EBM machine into North America. And we were down here in New Zealand and we were sort of solving interesting problems, making custom surgical instruments for local surgeons. And there was a real need to make implants as well to help some of these weird and wonderful cases that we had. So I found myself sitting at a desk sharing an office with Paul, who you can see here, creating what was really some of the first in man applications of printed titanium implants. So what you can see on screen is a prototype of an implant for a maxillary sinus tumor that was removed. And when you start to think about what are the amazing sort of creative applications of 3D printing, you know, this is 15 years ago now, we were already reconstructing a quarter of someone's face with a piece of printed metal. A year later, the implant side of the business was spun out of Instech and into another company called Osis, which some of you might have heard of. I'm going to show a nice gory photo now about some of our early work. I won't keep it on the screen for too long. So Osis was really a vehicle to say, what problems are surgeons having and how can we use printed titanium and the creativity that us have to think about, new geometries to solve problems. What you're looking at here is a pretty substantial femoral augment. So this is someone's knee that's bent over and they've had a bone tumor removed. This is actually the patient's 8th revision surgery. And so this is just an absolute nightmare situation for both the patient and the surgeon. And we created this really quite huge augment out of printed titanium scaffold. And the idea was that, and you can't just fill this up with bone graft because it's not really going to provide any structure, but we can simulate some of the properties of bone in terms of its structure using this titanium, and we can put it in there and it's going to provide a lot more mechanical strength than morcellated bone. And it started to work, and we started to get traction with some of the surgeons that we were working with. And so this first application of this technology then led to a bit more focus around, what's some of the, I guess, killer applications for AM. And we ended up making products like you can see here. On one side, you've got a relatively normal hip replacement. It looks sort of like all the pictures that you see with a femoral component and an acetabular cut. And on the other side, you've got this monstrosity, which is a fully custom 3D printed titanium hip replacement. And the need for products like this comes out of, I guess, not your routine surgeries. This, on one side, you've got a routine hip replacement. On the other side here, you start with an implant, and then the tissue around that gets infected, gets lysis. You have to remove a bit more of that. You have to revise the implant in the process, and you end up with less and less bone stock as you go along. Other cases like this, you might have removed a tumor, and so you're left with significantly reduced bone stock. And so Osis's core business began to focus on these custom hip replacements. And fast forward a year from here, I found myself doing my PhD working out how do we get these implants to stay attached to the patient. And so I found myself standing in a paddock in Lincoln, looking at a flock of sheep, hoping to hell that all of the money we spent on the preclinical trial worked. And that's a really interesting situation to be in, because you end up spending a whole bunch of money on, I guess what you'd call good basic science, good basic research to prove something which you kind of think you already know, but it is actually in the process of confirming that it works, that you're adding value there. And this is me, and this is now my business partner, Brent. Brent was along for that journey as well. He was a freshly minted board certified small animal surgeon, so the equivalent of a consultant orthopaedic surgeon in human, but for animals. And so we brought him into the project to provide some expertise around the application of this technology in a preclinical setting. But Brent really asked me the question. It's one of those painfully simple but cutting questions of, can we use this technology to help animals as well? We use animals in research all the time, particularly at the business end in terms of translation. And Brent's point of view is, well, it would be really nice if we could use that same technology to help animals as well. The answer, unfortunately, was it worked really well. And so we kind of didn't have any more excuses. We proved that it worked. This is a histology slide of a sheep's tibia, so a cross section through just under the knee. And in black, you can see the titanium implant, and in green, you can see that bone growth already starting to grow into it even after just 3 weeks. That then led on to some pretty substantial funding from MB and a couple of industry sponsors with Tim and at Otago there. And so we had sort of just over $3 million to continue on with that research into the next application areas. And so we were pretty confident in the technology there. And so Brent and I's partnership went to that next level of setting up a company. And so from 2013, we set up Possibility. And the purpose of that was dogs get sick just like people, they get injured just like people, but the treatments that they were offered were less effective than they could be. And our goal, I guess, is to overcome that by closing the gap between veterinary medicine and human medicine by bringing those two technologies together. So a little bit about where we are today. We started to think, okay, we've got this great technology, we can do these custom implants, but is that really a business? We had a first couple of customers who spent a heck of a lot of money on their dogs. You know, when people spend $25,000 to fix a dog, you take notice of those kind of numbers, but those were few and far between. And so we really had to say, what are the most common orthopedic conditions that animals were facing, and overlay that with what techniques had the greatest opportunity for improvement using AM. And what we found, I guess, was a problem worth solving. And so cruciate disease in dogs, just like in people, is one of the most common elective orthopedic procedures. In fact, it's so common that every year 1 in 100 dogs throughout the world will get treated for cruciate disease. So that's a multi-billion-dollar market that I guess I knew nothing about before I started looking. And we built this really simple product. What you can see on screen here is essentially just a wedge of titanium scaffold. It's a really simple product, but it hit the surgeon's biggest pain point. What this product allowed us to do was to cut and reposition a bone and then simply put in place this spacer, which held the bone around it in place during healing. It facilitated the rapid fixation of these two bone fragments to join together and heal, which gave that patient a much faster recovery, much faster return to function, and it significantly reduced the complication rate around mechanical failure of these constructs. We learned pretty early that we needed to support that product with things like cutting guides, what we call decision support, so pre-surgical planning and education. So what you can see here is our osteotomy guide, and this allows the surgeon to make reproducible cuts. It's what allows us to take an X-ray of a patient, to plan a surgery, and then to execute that surgery on that patient. We learned a lot from those techniques, and so a few years ago we launched our first printed instruments as well. And so what you can see here is a slightly different technique, but a cutting guide, which is, you know, essentially just a couple of dollars worth of nylon printed out, which allows a surgeon to make a very accurate, reproducible surgery, which, you know, prior to that, they were doing freehand. So it's really just one of those examples of, this would be a very expensive product to produce in metal. It'd be a very expensive product to produce with traditional manufacturing, but it's something that was really cheap and easy to do with 3D printing. And so it created a lot of value for that end user. From there, we have moved more into the planning space. And so we have a pretty comprehensive in-house surgical planning process. We employ computer scientists and all sorts of things to help us take that record of thousands of patients and thousands of x-rays and turn that into intelligent algorithms, essentially, so that we can plan better surgeries. We can say dogs like this who got better were treated in this way. And so we can start to draw out some of those lessons learned and to do things in better ways. It's also not just about having products, it's about having a complete offering as a business. And so things like workshops, education for surgeons are all part of us learning that we need to offer a complete package, a complete solution in a space, rather than just having a nice shiny widget, I guess. And so over time, we've been able to grow from a tiny little company that had an idea to use this guy's PhD project in a dog down the road at the clinic, to really having a healthy business. Every day we come to work, we have between 3 and 5 surgeries that happen in New Zealand using our products. Dogs walk in with a limp and they go home without one, and that's a pretty special thing to be a part of. And it's pretty amazing how such a, I guess what we call now a mature way to build things, can have such a transformational impact on patients. So I think with a couple of minutes remaining, what I'd like to do is just show you the end-to-end surgery for this procedure. So if I try to do that, so I'm here. Alrighty, you should now be able to see. Can we all see a video there? Right. Now we're gonna. All good? Increase that. All right, so that's us. Great.
Thank you, Seamus. That was actually really awesome to hear the story from end to end, because I know we've had many conversations in the past and it's been snippets. I guess one question I would have is, before I open it up to everyone, just being selfish here, what you've done in the veterinarian area has been fantastic and it's really sophisticated. Do you know how far we've come with humans?
Absolutely. So I'm still very involved on the human side as well. So yeah, for my sins, I also did a postdoc at Otago. So Tim, my old boss, is on the line here as well. And it's amazing to go from where we were back when I was doing my PhD, doing, being really excited about this new technology, to then saying, can we use this in more interesting ways? Can we make, mechanically sensitive devices? Can we use this in different application areas? And actually see that translate into products in patient. So some of the work with OSIS, those ideas that we were playing with only a few years ago, running preclinical trials, are now, spinal fusion products in patients making a difference. When we started, there was no regulatory framework in the US for additive manufactured medical devices. Where we are now is we're on sort of a second and third generation of regulation around manufacturing processes, around what I'd call a box rule for custom implant designs, saying, here's the range of possible solutions and our product will exist within that. And really, with the increased framework for regulation, it's accelerated the delivery of printed products. It's a real happening thing. If you're going for a hip or a knee replacement in particular, there's every chance that that'll be, if not a printed implant, then printed cutting guides. And I guess my side of that is when you take your vet to the local clinic down the road, there's every chance that they'll be receiving the same technology.
Okay, thanks. It's really good to hear, and good to see that it's actually accelerating and being accepted now. Aidan, you have a question.
Yeah, thanks. That was awesome, Seamus. I'm not that familiar with the regulatory kind of oversight in the animal, animal and veterinary clinics. Like, how difficult is it to enter the market in that space? Is it comparable to, a medical device for humans, or is it lower?
The short answer is it's lower. The slightly longer answer is that, a little bit tongue in cheek, but it does cost millions of dollars to go through a regulatory process for human devices. And the difference is that in the veterinary side, you need to have a pretty good war chest behind you in case you get sued, because whilst there's no approval process, at the end of the day, you're still putting products into patients and they need to work. And if they don't work, there's going to be some pretty unhappy customers and some pretty unhappy owners. And we're looking forward to going into the US with this product June next year. But with that sort of comes some different challenges there. It'd be really nice to just say, hey, look, this is an FDA approved product and it's on you. But the reality is that we have to satisfy ourselves that these products do what they say on the tin. And so there's still a lot of testing and proving that they work that needs to happen.
Yeah, okay. Thanks, Seamus. That was very helpful.
Yeah, thanks, Seamus. I think we've got Alfredo with a question, and then Masako.
Hi, Seamus. Thank you very much. That's quite interesting. So my question is more around something that you touched on earlier. Can you all hear me?
Yep.
So what you've done is very smart. You have not tried to do N equals 1, right, of sort of printed implants, 3D printed implants, because that creates a huge amount of issues when it comes to reimbursement or getting paid by government and things like that. And that way you don't have a sustainable business, because there's only a handful of people that can afford a $25,000 dog implant, right? And we see that in the clinic as well, in humans, right? That there has to be a viable reimbursement platform, in order for these things the technology exists. It's just a matter of how do you deploy this as a business, right? And I think having a strategy where you identify a problem where you can start off with a generic sort of solution and then potentially customize that solution once you are within the surgery or after you've taken some scans or whatever. I think that's the way to go. My question is more around do you have any existing partners for the vet applications here in Australia? Because my dog may be a recipient of all this, and I don't have $25,000, by the way. So
Yeah. The way I look at it is, I mean, we certainly did start with custom products and learned pretty quickly that, yeah, not everyone has 25 grand to fix that. And it used to be that you modify the implant to fit the patient. I guess with routine surgery, you flip that on its head and you modify the patient to fit the implant. And it allows you to actually build a business on it. And at the end of the day, it has to work in a commercial sense in order to be able to help people or animals. Yes, we do have clinics in Australia. Actually, the majority of our revenue comes from Australia at this point in time. So yeah, there's every chance that the clinic that you go to down the road would be using our products. And if they don't, you should tell them to use Operability products.
Very good.
thank you. Very interesting. Thanks.
Thanks.
Yeah, Masako, you have a question.
Yeah, I guess just, following up from, Aidan's comment about the regulation. And I think, Aidan, Aidan's, presentation, you put, highly regulated and non-regulated in the research and sort of human. And I guess for you, Seamus, it's in, animal. But I guess, when you have a great idea, but I think for this 3D printing, bioprinting, tissue engineering, you guys are in the forefront of, new technology where even the regulation itself don't know what to do. Do you have any, I guess it's kind of open-ended, do you have any comment on that? Just because of the area that we work in, how do you navigate in that sort of, cutting edge, bioprinting, which I don't think even FDA, TGA don't have a concrete guidance how to go about it.
Yeah, so in our situation, and you've hit the nail on the head, the framework isn't always there, and so you don't know what the rules are. And my experience has been to lead into that and to get engaged with regulators to help build a framework that you're never going to build something that suits just your interests, but you'll build something that enables you to build the types of solutions, build the types of products that you want to be able to build. So specifically with plastic AM and metal AM, being part of regulatory meetings with the FDA, they have a lot of working groups, they have a lot of consultation, and if you have specific expertise in a field, they will listen to that. You might end up on a working group, which may not be your cup of tea, but you do have a lot of influence. And at the end of the day, they are there, the regulators are there to make sure that the products are safe and effective. And so if you're an expert in that field, they will listen to you and listen to your colleagues to help inform those regulations. And, it's taken 10 or 15 years for the FDA to put frameworks in place around this stuff. From my perspective, it's fantastic because it keeps the cowboys out.
So, sorry.
I was going to, the only question I'd come up with was in this same space, slightly differently, and I think, Seamus, you're addressing it, is even just in the few that I know, that the development just seemed to be moving so fast in this. And I was wondering how the regulatory agencies, not specifically FDA, I guess I should be thinking about TGA, do they have a plan for keeping up? I mean, your comments about, being involved and getting in there is useful. That just relies on you being vigilant and interested enough to do that. But I just wondered if, and there may even be someone in the audience, whether the agencies themselves have a plan for keeping up, because it just seems to be moving really, really quickly with really good benefits being shown.
I'll start off just with one comment, and then lots of other people have sort of more experience, particularly on the tissue side of things. But if you're the poor schmuck that has to do a PMA, that's a really expensive process, and you're going to be demonstrating, oftentimes for the first time, how these products should be tested, should be evaluated to prove the endpoint that you're trying to prove. Particularly with tissue products or scaffold products, it could be a combination of device and drug at the same time. And so you're sort of dealing with two different pathways there as well. So I think you do need to make a choice whether or not you want to be first or whether or not you want to be a fast follow. And sort of come in with a Me Too product, or like a 510K or something like that in the US. But it does tend to be an industry sort of push, I guess, and the regulations will follow, rather than the regulators having a master plan and you fitting into that.
I guess there's upside to that, and as you said, in having a bit more say and close to the driver's seat. But yeah, just looking at some of the things that Kuhn's doing, and even Aidan with skin, I just wondered, even Aidan, do you think your reg path forward will be fairly smooth, or do you see some bumps there you need to negotiate?
It's a great question, I think. Similar to what Seamus said, I think we're still working on our regulatory strategy for legal. It's a bit unclear because I think the regulations potentially don't exist for our product. It could be, depending on how we, what indication we land on and how we best see how to bring it to the market. It could be, in some scenarios, it could be a class 2 device, right? And we can be a Me Too device. There's another scenario where it's a class 3 combination device, right? Where you're, coming in as, the bar is as high as possible, and it's brand new, right? And it will get put under the microscope, I think. But so yeah, it for us right now, it's a little unclear. I think we're going to continue working on it along with the development of the technology and the product. So yeah, so that's about as well as I can answer that question at the moment, but we'll see how it goes.
Yeah, good.
Sorry, I was just going to make a comment. I was just wondering, Kuhn, with the bio inks that you've developed and you've licensed to Merck, did you have to go through any regulatory requirements for that?
So for the ones that we're doing, no. I was gonna say, like, when Lance was asking, is it gonna be smooth or bumpy? I mean, it's always bumpy. I feel like regulations it's always not straightforward because things change really quickly. And I think the FDA have the new regulations for the 3D printed personalized ones in November last year, and there was a number of working groups that went on to that as well. For the materials, I think the use that we have licensed them is for R&D only, so it's not for patients use. It's not in humans yet, so it's easier, and most of it is lab-based. But indeed, if you want to bring it to humans, then that's the whole pathway that we need to undergo as well. And especially with cells, I think it's definitely more challenging.
Yeah. So I was just going to put in a plug for the Kiwis in this audience, actually, and perhaps some of the Australians as well. With our Te Toki Mataora MedTech Research Translator Program, we do have a module which actually provides an upskilling in regulatory matters. So it's really just getting people to understand regulatory requirements and FDA requirements, and help, so that help you introduce you to some of our regulatory experts who can help you with developing your strategy going forward for your products. So at the moment, this is only open to Kiwis because it's face-to-face meetings at the moment. But we're actually thinking of running a workshop, a virtual workshop, which I think will open it through the BioBridge in case there's any interest from Australians. If you think that's a good idea, Lance and Alfredo?
Yep. It's an area that anyone developing any med tech has to engage with at some point, just like I've been saying lately, the need to engage with the public health system just for volume and expertise, and the regulatory stuff you can't get away from.
Mm-hmm. Okay. Do we have any other comments or questions?
Yeah, it's Roger here. Can you hear me?
Yep. Hi, Roger.
Hi. Those who don't know me, I'm Roger Warren. I work with the surgical engineering lab at Auckland. Yeah. Two questions. One for Koen. Resolution and speed of printing, things like that. Can you comment on what your resolution you're achieving with your 3D prints?
With the lithography printers that we have, we can get up to 25 microns on some of the hydrogels, depending on how well we can optimize our materials. And with the DLP, so with the lithography-based smartphone printer, for example, we were able to print about a 5 centimeters construct in less than 30 minutes. So that's sort of the speed that we're trying to get, because once we have cells in them, we want them to not be undergoing the printing process for too long in case of we're going to jeopardize the cell the cell viability.
Yeah.
But it does come in finding the balance between, is the resolution that important? Because if you print it way longer, your cells might be less functional, whereas the functional output is what we are more interested in, mostly in terms of making these tissue constructs.
Yeah, that's what I was coming to, is like, also with the resolution size, are you seeing that certain tissues are getting suit compatibility with the drop size or your resolution size? Or are you really optimizing for time and getting a very big resolution?
It does depend on the application. So it's like what we're trying to make. So for example, for all of the blood vessel stuff, the microcapillary stuff, resolution is then really important, because you want to be able to get the resolution, but then the functionality is then to be able to perfuse the microvessels that you form so that it behaves slightly similar to blood vessels. But most of the time, like bulk constructs, it's less. Sometimes shape is important. With a lot of the decellularized extracellular matrices where, mechanics is important. So you need to get the right overall, I think, mechanical microenvironment to support the cells to differentiate down the specific route. So I guess, like, short answer to that whole long waffle is really balancing all of the different parameters hasn't been super simple, and it is very dependent on what application you want and what cells goes into them. And there is difference if you put stem cells or already mature cells. But I think as a whole, we have always been looking at the final function. So I think tissue function is more important, because if you print something and has a beautiful shape but doesn't have the function, that also is not quite useful in the tissue engineering scheme.
And one more question for Aidan. Aidan, we're working on a chronic wound device, and I can see the skin tissue production you're making is very useful to our research. But also, when we have a product that comes out of it, it's a very good add-on product to, once you've set up tissue, how you then continue to keep that healthy over a period of time. So I'm wondering if there's any opportunity for a collaboration with what we're doing. We're early days yet, but we've got some good indicators. We're going to have a good product at the end of it.
Yeah, definitely keen to look at anything that's synergistic that we can collaborate on. We typically collaborated with a lot of different folks and universities here in Australia, but keen to explore. I think Lance probably has my details that he could probably share with you afterwards if you want to get in touch, and we can have a chat about it.
Good one, thank you.
Excuse me, Roger, you're from the Surgical Design Studios, right?
Yeah, Surgical Engineering Lab under Professor Anthony Phillips.
Yeah, okay, cool. I know where to find you then. Tim, you had a question.
I did. Thanks. Thanks, Guy, and thanks everyone. I was trying to think up a question that would be relevant or pointy to all three panelists. I think I have one. Before I ask, I was just also going to say that I think it's that nice discussion we're having before around the regulatory ones, because I think we're all at that sort of nexus between how we can manage our automation or printing technology that meets the application and that the R&D space, even for, Inventure and others in the R&D space, in terms of being able to have, high throughput is the driver and the simplicity. But then we're also trying to go, right, well, how can we take that then when we go into the human case, and understanding what the regulatory pathway is. And again, that approach around going towards single surgery that Aidan was trying to talk about, what can we deliver in surgery, again, ensuring that the cells don't have to move away from the surgery to then, to have them delivered back, or what can we provide that's off the shelf. So I think, that's very much the sort of nexus where I think the whole field is at, in terms of both the technology side and then how you're managing the solutions for the clinical side that are really going to try and make sure that you can navigate that bumpy regulatory pathway, or there's some clever things that you can not necessarily make it easier, but you can streamline that process, which removes some of the barriers. So I think, that's something that I think there's been some good examples talked about today. What my question was for the three panelists was actually, I think, one could look at printing in medicine and think, oh, well, if I've got then a fancy printer, or I have some printing technology that's essentially solved like 90% of my problem. All I need is a printer, and then I'll be able to make something. But I was keen to get the thoughts of the three panelists to say, right, well, in reality, then how much is then the understanding, or could you give a quick indication of how much value there is around understanding the actual manufacturing process, understanding the materials and the development side that has to go into that process? So give an indication of what that weighting is to try and that allows you to have this, I guess, sort of success that you have in your applications. So yeah, I guess my question is how much value is there in terms of understanding the steps of your process and the actual manufacturing process, as well as the materials that go into that process, as opposed to just the technology on its own.
I can tackle that first. Hey, John.
Thanks, John.
From the very early days of what I was doing, you are part of a team, and each person within that team is an expert in their domain, but you need the whole team to play together to deliver a solution in this space. So I'm an expert in the design for additive manufacturing and the application of it. I'm supported by people who are experts in the materials. In my case, they're metallurgists, they're, polymer experts. That's also supported with people who are experts in building these machines and making them reliable. And then on the a bit further down the line, I'm supported by people who are experts in the clinical application and driving product choices from what do I need to help solve this problem sort of perspective. So you learn pretty early on that no one person is going to span all of those roles. And I think it's important to recognize this is the bit that I'm going to be doing, but I need to surround myself with these other people to complete the puzzle.
Yeah, maybe just to add on to that, I completely agree with Seamus's point. And you can't really span these disciplines. I think one person can't really span these disciplines, and if they have, then I haven't met them yet. Maybe they do exist, but like for me personally, I had no background in biology before starting Inventure, right? Like I'm an engineer, and I know a lot about printers and printing systems and designing products. But from the early days, Inventure grew out of an ARC linkage grant with UNSW, where we had Professor Justin Gooding's group developing the biomaterials and Professor Maria Cavalaris's group doing the biology and the application. And it's that meeting of those minds and a meeting of those disciplines all working together, and we've tried to keep that at Inventure as we grow. We typically do all our work in cross-functional teams, where you have biologists and scientists working with engineers and material scientists all working together to try and deliver something that will hopefully make our customers' lives a little easier, and one day kind of hopefully help patients. So yeah, I guess the other thing, from a technical perspective, is you can't really everything has to work in kind of symbiotically. You can't just develop a biomaterial without thinking about how you're actually going to deposit it or, engineer what comes out of it. It's all got to be designed to work together. And I think if you try and design different pieces of a system in isolation, you end up with something that doesn't really work very well.
Following from Seamus and Aidan, but I don't think we probably know this already. I mean, you do sit next to me in the office. It's mostly really, I think, from a research scientist point, I feel you will know more of what you do. I mean, my expertise is in photopolymer chemistry. I'm a material expert, so I know a lot more of the bioinks. I do think you need to know enough though of the other components, so not completely oblivious, not knowing what's happening. You do have to know, how the printer's behaving and all of that. But then, of course, a lot of it comes down to assembling a good team, and then you have partners, you have collaborations, and you trust them, believe what they say, because you can't also just make sure that just everything that they say you blindly just follow. But knowing enough, but then also building that trust, I think is key of making the whole thing works.
Just to add to, just to add to that.
You go, Seamus.
I think knowing enough about each of the different parts of the puzzle is really important. Thinking about that, I kind of call what is knowing enough about an aspect of something. If it's critical to your business and critical to the end application, then I'd kind of define enough as a PhD in the technical side of it. But the reality is that even then, that's only going to scratch the surface of all of what goes into what ends up in a patient. So yeah, these are complex products. They're deep tech products, and they need a lot of expertise.
I guess at the end of it, with the regulatory side of things, it's really about risk management. And so, there is a risk in the printer that you use, so that needs to be consistent, the bioinks that you use, the method, and finally the product that you come up with. And so if you have got all of that risk management bits in place, that possibly helps you with some of, maybe that helps with some of your queries or uncertainties. Anyway, that's just my perspective. But Alfredo, you've got another question?
I just wanted to make a quick comment on what you just said about regulation. So for those that are not familiar, what TGA is looking at in Australia is to have a design envelope, where within that envelope, which includes the printers and the powder that you use and all the things that go into making the product, if you are within that envelope, right, then it's straightforward that that product gets the tick of approval. Yes, you can use that product, right? Because it's going to be impossible under any framework to have every single step, in an N=1 validated and looked at by the regulators in each case, and have an approval within a time frame that is going to be, useful to you, right? So it has to be something where we agree on a design envelope. There's all the quality management and all the quality controls within that design envelope, and then you basically submit it, press a button, and this thing gets, submitted and gets approved, and then you can go on your merry way. We still need to deal with the reimbursement, which is the business side of the equation, and that's an ongoing process. But I think the regs are almost there, right? I think that's pretty much settled.
Okay.
I guess if I can add one small comment on that. Oh, sorry. I think there's two of us all.
Go, Tim. You go, Tim.
Oh, sorry, Lance. I think the point around the regulation and also your risk management, Di, is true. I think we can learn a lot from what we've understood, is that actually, developing of cell therapies and that regulatory pathway has been difficult because, they've evolved together. But what we know hasn't worked, particularly in the cell therapies or tissue engineering space, is that you can have a product that could be regulated and approved, but again, the cost and the time it takes, it's just astronomical. You never end up with a viable product at the end of it. So at the end of it all, while you've gone and ticked all the boxes for the regulatory pathway, essentially, it's a dead end from a commercial aspect, or it's so expensive, like I say, you'll never get the reimbursement or insurance. I mean, there's plenty of examples in terms of how that went, I guess, in the tissue engineering space. And so I think, this is where this is nice nexus is that we've got sort of some things where we can automate things to simplify. We've got further developments in how we're getting allogeneic cell sources available and a few more things off the shelf. So it doesn't necessarily solve a lot of things, but I think that's where I think the field is at, and there's lots of opportunities in that space. Still means that there still needs to be that regulatory pathway, but got sort of gone are the days around having these processes are complicated enough without then trying to come up with a construct that's individualized to the patient that might include cells. And it's difficult enough just to have a custom titanium implant which doesn't contain cells at all, right? So I think it doesn't mean that there's there's not further opportunities there, but I think that's sort of a really interesting intersection at the moment between our materials and our manufacturing, and again, sort of the R&D space versus then also what we can get to into the clinical space as well. I still think there's a lot of advantages in terms of what we can do in the screening space and the R&D, which will provide a pathway to then how we're able to make maybe slightly larger functional tissues or implants that can get into patients. So anyway, that's just my 2 cents worth a little bit around those discussion points.
That's a good 2 cents worth. 3 cents, 4 cents easily. A question for Aidan. In your presentation, when you compared, what LEGO is going to be doing to, what happens today, there was a step where you obviously need to harvest some skin cells to put into the, but you also come, that today that taking of skin cells can be more painful than the wound itself. Is there an optimum? Obviously, to get maximum benefit, you want to have minimum number of cells that you take. Is there an optimum number of native cells that you need to take to put with the matrix or the ink, or it's just some?
Yeah, good question. I don't think there's enough data or evidence to know the exact number of cells per area or volume at the moment to really understand what's the minimum you need to trigger that regeneration. But if you take the example of RECELL, the spray-on skin product that's on the market in the US, was FDA approved in 2019, I think it was. What they quote is I think 80 or 100 to 1 ratio between the wound site and the donor site. So that's a very, you're taking a very small donor site to treat quite a large wound. And typically in a split thickness skin graft, I think it's about 3 to 1, right? So you need to take a skin graft that is 1/3 the size of your wound. So it's can be, if you've got a pretty significant wound or a burn, the donor site can be quite large. And it also means as well that, and particularly in the case of burns where you've got, very significant percentage of your skin, like in a chemical burn or a fire, you don't have that much healthy donor skin, right? If you're over 50%, then, there is not that many places you can actually take skin from. So it's an interesting challenge. And I think, I don't think we know exactly yet how much cells are available do we need in the matrix to actually, trigger that regeneration phenotype.
But triggering regeneration and time as well, I guess, becomes one of the tricks you're looking for. Yeah. No, thank you. Any other questions? No. Time is it? Oh, might be finishing early, Di.
That's okay. I mean, we always have this for 2 hours, but it's kind of one of those where it goes for as long as it goes or as short as it goes. But I think we've had some really good discussion today, and some really good presentations as well.
Yeah, no, great. Look, thank you to Aidan, Seamus and Koen for joining us today. I think there's at least one connection we want to make between Aidan and no, I can't even see the name because you're all in the room. But Di knows who you are. She said she'll track you down.
Yes, Roger.
Yeah.
Yeah. There might be also another connection for Aidan with someone at ABI who's working on needle-free injection technology that might be able to be used for extracting cells less painfully.
Anything that's less painful sounds good to me.
Yeah.
The same thing.
Mhm.
But yeah, so thank you again to our speakers today. There are no other questions, comments, then yeah, we'll call the workshop to a close. Thank you everyone for joining us. Thanks Di and Ella and Kate at your end for organising things today. But if anyone has any questions or comments or want to be connected to anybody after we close off today, just get in touch with Di or myself and we can sort that out.
Yeah, and also the other thing is that I know everyone was here for the 3D printing in health workshop, but if you have any other topics that you would like to see or hear about, just reach out to us as well, because you know, what we like to do is actually curate workshops around what the community actually wants to hear. Okay,
Good
cool.
point. Thanks everybody.
Thank you, everyone.
And thanks
Thank
again to our
you.
speakers.
Thanks
Thank
so
you.
much.
Bye.
Thank you.
Bye-bye.